Hello and welcome to the March 2025 Mission Impact Volunteer Call on Cell Therapies. Today's presenters are Esther Latris, PhD, Nicholas Mamrak, PhD, and Mariana Marnak, PharmD.
Hello, everyone. I'm really excited to present you an update on cell therapies and all the advancements from a year ago. So just to remind you that cell therapies is key to our mission of advancing breakthroughs to cure, prevent, and better treat people living with type 1 and complications. So the objectives or the strategic initiatives of the cure programs include cell therapies and the disease-modifying therapies. You may notice that we had an early disease and prevention and screening portfolio. Now it's all incorporated into the disease-modifying therapies. And the main focus and objective is to delay and ultimately prevent T1D. You're familiar with the Improving Lives initiative, including device, drugs, psychosocial complication, and global responsibility. We are working very closely with business units, with research, with advocacy. It's a new team of medical affairs that I will introduce later, and as you're familiar with our partners and T1D Fund. So I'll give a little bit of overview, and then I will have Nick talk about the portfolio, which is an advancements with it's the most important objective of this call. And also Mariana will talk about the regulatory and advocacy part on efforts that we're doing in the cell therapy portfolio. So you've probably seen this slide, but if you haven't, this represents our vision of a world without type 1 diabetes. So on the left side is a representation of the functional mass and how decrease in functional mass is linked to the progression of type 1, and also what are the different therapies and approaches that we are focusing at different stages. So just to remind that stage 3 is where the clinical manifestation of type 1 diabetes, it's manifested by a need for exogenous insulin, but we are working very strongly, and we had already the presentation on early detection in stage 1 and stage 2. Stage 4 is beyond the new onset, and that's where we're going to focus on. And the main two portfolios and strategies that we have, it's a cell therapy portfolio and also the treatments and improving life. So this slide, I think you may be familiar too, but it represents what really breakthrough T1D focuses on, which is taking therapies and breakthroughs and discoveries from very early on to access and adoption. So at the top are the main two pillars of our objectives. One is advanced breakthroughs that goes all the way to a regulatory approval. But as we all know, regulatory approval is not the end of the road. So the unlock access are all the activities that we are focusing on increasing coverage of these therapies. These therapies, once they're approved, they need to be adopted, and this is part of our focus, making sure that it ends up in the hands of people with type 1. Access and adoption is mostly focused on supporting continuing healthcare provider education, making sure that healthcare providers know about these therapies that are coming. And also educate the community on clinical trials, on new therapies that are coming in, making sure that all these therapies are adopted. So we're going to talk about cell therapies, and I think I just want to frame what the cell therapies are. And on the left side, there's this islet with different colors that represent the different cell types in islets. I just want to point out that when I talk about manufacturing beta cells, we are manufacturing actually an islet. We're manufacturing now islets that can contain different cell types. So they're working like an islet, like endogenous islets. But the blue one is it's what secretes insulin. The beta cells are the ones that are more susceptible to the immune system. We're studying maybe why the other cell types, they don't induce or autoimmunity. So this is something that we're still focusing on the discovery side. So cell therapies place insulin-producing cells back into the body to provide insulin independence. And that's the main goal of cell therapies, is providing insulin independence. The focus areas that we are pursuing can be summarized in 3 areas. Cell source. We need a cell source that can sense changes in glucose, that can provide insulin in response to changes of insulin. Cell survival. How are these placed into the body in a side or that can provide vascularization? So obviously these cells need to be close to blood to sense changes in glucose. Longevity, how long can these cells last? And finally, the third one is protection, which I think, it's one of the main challenges, is how can we protect these cells from the immune system? So we do have an approved cell therapy. This is a donor islet transplantation into the liver with immunosuppression. So if you think about the cell source, in this case, our donor islets, cell survival is the liver that's been shown to provide a good environment for these cells to survive for many, many years. And cell protection, how these cells are protected as any other organ or islet transplant, is with chronic and systemic immunosuppression. So our goal here is to basically provide a new source of cells that can be manufactured, find additional sites where these cells can survive and can function for a long time. It's the liver, but we may need some other sites. And we talked about the omentum, and we talked about the sub-Q sites. These are subcutaneous sites, the skeletal muscle. These are other places where these cells can be transplanted and survive and function really well. We have some evidence of that it at least can survive and function for some time. So, and also the other main challenge is immunosuppression, or the way the immunosuppression it is now. So it's the whole thing, it comes to a benefit risk ratio. It's the current immunosuppression has some risks, and it may be beneficial for a population that now it's the severe hypoglycemia or the hypoglycemia unawareness population. But we are trying to, there's a lot of data on the immunosuppression, the safety of immunosuppression for a long time, that could potentially be expanded. And then Nick will talk later about some of the things that we are funding to make immunosuppression, or find new immune suppressive therapies that are less toxic. So very fast manufacturing islet cells. There's been an amazing, I think, great progress in the last 10 years. Scientists were still figuring out how to make these cells precursor cells. This is a cell that can go in many different directions, has the capacity to form little organs and cell types. So how you direct these cells to become a manufactured islet there's been a lot of progress, and we know now how to do that. We're still working on some differentiation protocols, and most importantly, how to manufacture that at high scale. We've done a lot of studies in what's called non-preclinical pharmacologies and some laboratory animals, but we need to definitely look into making millions and millions of these cells to meet the clinical demand, durability, and obviously the safety. While these cells, most of them will become endocrine, some cells may not see the signals that they need to respond to, and that's something that we need to look into as well from the safety point of view. Immune protection is it's where we're putting most of our effort. So encapsulation has been a long time. It's been a long time research effort, even in the clinic. We still think it's a valid way of protecting these cells. I think one of the challenge here it's been the predictability of these animal models to humans. And I think while there work really some of these have worked very well in animal models and the predictability into humans, but we're working on that. We're working on maybe putting some coatings on these biomaterials, finding better biomaterials, or maybe implanting them in sites that may not induce a lot of what we call fibrotic response. The second one is target immune modulation. This is based on protecting the cells from the immune system. This would be no membrane, but a way of having these cells, agents or drugs around them that can prevent the immune system from recognizing and attacking the cells. And the third one is the gene editing. So can we make these cells smarter in a way? Can we, as any cell that we put in our body that comes from a non-self, it has some red flags that triggers the immune system, and the immune system doesn't recognize as a self and it gets rejected. So can we make a smarter cells that do not trigger this immune response? So obviously from the little introduction, you can figure out that not on one person on one lab can do that. So it requires beta cell biologists, bioengineers on biomaterials, clinicians, industry manufacturing, cell engineering, and immunology. So for a long time, we had the beta cell replacement consortia as a way of bringing all this expertise together. The cell replacement consortia has evolved. It used to be very involved on the beginning, on discovery, on testing biomaterials, on protocols for making islet cells. It's now evolved to a place where we think about products and we think about what are the products that we need to move forward. And obviously, the large pharma industry has made significant advances, but we still, as Breakthrough T1D, we still need to bring more innovation, more ideas, more prototypes that maybe haven't thought before. So this is an area that we are definitely focused. And another strategy or another focus that Breakthrough T1D has explored and significantly now successful for the last few years is bringing these centers together, is providing opportunities for new research, for collaboration, for training young scientists, for taking a risk, for providing funding and longevity of projects where scientists can focus on taking some risk. So this is just a summary of the active grants. So these are the grants by projects on the left side on the different areas of research. So cell therapy is about 25%, 32%. I just want to make sure this is our grant portfolio, but also there's the resources outside Breakthrough T1D that they bring a lot to this portfolio. So this is just the grants from Breakthrough T1D, but there's a lot of much that we try to obviously leverage from these areas. And these are the cell therapy grant pipeline. Obviously, there's a lot on early preclinical, and we would like to see more late preclinical and early clinical. And this is part of the Project Act that I'm gonna advancing cell therapies, right? If you look at the discovery and early clinical, the more we advance into early clinical, the costly the projects are. Clinical trials are much more expensive than maybe a discovery experiment or study. So the resources that we need to put into have to increase significantly, and also resources from Mariana is going to talk about regulatory or the medical affairs. How do we prepare the field, and how do we position these therapies when they're going to be ready, which they're coming? So this is part of our Project Act. It's just not reinventing anything, but just bringing resources. So this discovery, 39% can advance faster and much more efficient. No, it is still efficient, but we still need to put more resources. So we'll start, I think, going into the portfolio. I just want to introduce the recent advancements in cell therapy. I think it shows even in the last 4 years, right? And we don't need to go into details. We know Vertex or Nova, Lantidra, which is the first approval of the cell therapy, Eledon, testing now in clinical trials, therapies that are less toxic, and and the Sana. So the Sana's, Nick will talk about it, but the first recipient of islet cell that's been gene edited. And it's been at least it's only one individual. It's been it's in initial stages, but without immunosuppression. So I think this is very exciting. So this is just to show how much advancement has it been even in the last 4 years. So I'm gonna now move to Nick to give you an update on the portfolio.
All right, great. So my name is Nicholas Memrak. I'm a scientist here on the team working with Esther and Mariana on cell therapies. I'm going to be jumping in to give an overview of some current clinical trials and some select projects from our portfolio. So here you'll see sort of a snapshot of our pipeline of different clinical trials that are ongoing. There are multiple promising therapies in this area at the clinical stage. This probably doesn't capture everything. And I'll take time to go through some of these examples, give a little bit more detail on each one. In the interest of time, won't be able to cover everything. But just to note, within this list, we have trials both from industry partners in academia, and each one is really focused on one or more of these priority areas of either cell source, cell survival or delivery, and then immune protection. And so I'll go through a couple of these examples in a little bit more detail, and each time I'll try to relate it back to one of these 3 areas. So where we're getting cells, where we're putting them, making sure they're happy, and then protecting them from the immune system. All right, so the first example I'll go into is Vertex's VX-880 program. VX-880, or Zimislecel, is a manufactured islet cell therapy. So thinking back to our current clinical islet therapy, where we take cells from donor-derived pancreas, transplant them into the liver, and protect them with immune protection via broad immunosuppressants. Here we're going to swap out that cell source for a manufactured cell product. This trial is ongoing and very excitingly recently expanded to include a phase 1, 2, 3. And what that means is that this is the data where we're starting to get more patients. We're going to use this to look at both safety and efficacy to then seek regulatory approval by the FDA. And so right now, out of the 12 patients that have received the full dose of cells, 11 of the 12 have either reduced or completely eliminated their need for external insulin. So this is the first time that the therapy using manufactured cells like this has progressed to a pivotal clinical trial for type 1. And although this trial, this therapy does require immunosuppressants, it sort of is one step along the way of our pipeline for getting these therapies to larger populations, right? So initially, maybe starting a little bit smaller, looking at individuals with things like severe hypoglycemic episodes, but eventually, as we move towards other protection strategies, being able to open up that inclusion criteria. And a lot of this work, particularly this process for making the manufactured cells from precursors, was developed through breakthrough T1D support dating all the way back to the year 2000. The next example I want to dive into, very closely related, also from Vertex, VX-264. And so now this is using the same cells as the last product I described, but now instead of having cells into the liver, they're going to be delivered in an immunoprotective device. And that device is implanted below the muscle in the abdomen. And what this device does is create a protective barrier so that immune cells can't come into physical contact with our transplanted beta cells. Because of this, excitingly, the cells do not require immunosuppression in order to be protected long term. And so right now, this is a phase 1/2 study called Upward, enrolling adults with type 1, and some early data from this trial is expected by the end of the year. And again, sort of getting back to our different priority areas here, the need to have immunosuppression is really what limits the population for which these therapies are appropriate. And so using something like this device in 264 really opens up possibilities for who this could be appropriate for. Moving on to discuss another strategy for protecting transplanted cells. This example comes to us from Sana Biotechnologies. And this is an example of using primary donor-derived islets in combination with two unique technologies. The first is gene editing in order to reduce how likely the cells are to be seen by the immune system. And the second is a novel transplantation site. So instead of transplanting cells into the liver, here they're going to be transplanted intramuscularly in the forearm. We have some early data from this study. A first patient who received a partial dose of cells in this short phase 1 safety study has demonstrated that they are producing insulin from the transplanted cells, and there are no adverse side effects from the study material. And so this is some early data. This is just about 4 weeks after the cells were implanted. But again, this is in the context of no broader immunosuppression. So really it's that gene editing that's protecting the cells here. Now, this is important because this strategy of engineering islets is one of our go-to shots on goal for protecting cells. And this provides really exciting proof of concept data that an approach like this can provide protection without this immunosuppression. Long-term, the vision is then to apply this strategy to similar manufactured islets, moving away from the reliance on donor-derived cells from pancreata. And so Breakthrough T1D supported early investigations into this idea that gene editing might be enough to protect cells from the immune system, dating back to as early as 2015, 2016, to kind of look at early discovery research on this approach. And now fast forward all the way to today, the T1D Fund is an investor in this company, and we remain in close communication with Sana. All right, so the next example comes to us in the form of an immunomodulatory therapy. This is an asset from Eledon Pharmaceuticals, and it's a therapy called tegoprubart. This is a therapy that targets a particular molecule called CD40 ligand. And what that does is dampen the immune response and limit immune cell communication. Eledon had demonstrated the efficacy and safety of this therapy in the context of kidney transplantation, and this new study evaluating its effects in islet transplantation was supported by Breakthrough T1D. Taking some early data from the first few patients, the data suggests that this is potentially a less toxic way to keep the cells safe. So compared to what we normally do with our current standard immunotherapy or suppression protocols, less side effects means that we could potentially kind of shift who a therapy like this would be appropriate for. Breakthrough T1D funded both this initial study as well as the expansion to include more patients, as well as the T1D fund making investments in this company. Continuing on, this example looking at Cernova Biotherapeutics. Cernova is developing a method to deliver cells subcutaneously, so under the skin. They have a device called the cell pouch, which has been transplanted into 12 individuals and contains donor-derived islets. So thinking about, in this case, not the immunoprotection, but how we're delivering and keeping the cells surviving. So rather than being implanted in the liver, they're going under the skin. there were a couple different cohorts split up based off of how many cells they got or the design of the device. But out of the 6 individuals within that first cohort, 5 remained insulin dependent, anywhere between up to 6 months to over 3 years at this point. And so this is sort of still in development, looking at additional cohorts, but this is sort of an exciting demonstration of cell survival at this subcutaneous site. So Breakthrough T1D has supported this company, Cernova, and we continue to work closely with them for their next steps. And the last clinical trial example I'll give is from a company called Ceraxis. This is a new player in the manufactured islet cell therapy space, and is really right at the beginning of their clinical program, which is really exciting to have another entry into this arena. Their product, SRO2, is an investigational manufactured cell therapy. And this received FDA clearance for a new drug investigation for their phase 1/2 clinical study just recently. And so in this study, the cells, again, coming from a manufactured source, is going to be delivered into the omentum. And so the omentum is a protective layer that's found around the organs, and has been demonstrated in research studies to be supportive of transplanted cells. So again, thinking right now we put them in the liver, here we're swapping that for this new site to try to increase their survival. And in the context for this clinical trial early on, still protected by immunosuppressive drugs. This is exciting because one, of course, a new player to the space making manufactured islet cells. More shots on goal is always great. And then additionally, this is the first time that a manufactured islet cell therapy like this will be transplanted into the omentum, sort of validating that transplant site. Additionally, Ceraxis is also developing a modified version of this manufactured cell line, which will hopefully allow for treatment of this using this therapy without the need for anti-rejection or immunosuppressants. Currently, the T1D fund has invested in this company, Ceraxis, and towards their clinical trial program. A few more examples coming from our active portfolio now. One initiative that we've been really focused on is this idea of beta cell factories. As I mentioned in these clinical programs, a number of them rely on these manufactured cell sources, and that's really critical for getting away from the reliance on how many pancreases there are available for isolating cells. And so what these beta cell factories will do is allow us to have a consistent production of these manufactured cells at a large scale in a consistent way. And that consistency is really important because when we want to compare immune protection strategies, for example, head to head, having a consistent source of cells really makes that possible. And we're doing this by building partnerships with manufacturing facilities, including both the Advanced Regenerative Manufacturing Institute, or ARMI, and the Cedars-Sinai Biomanufacturing Center. Ultimately, this brings us closer to our goal of having a reliable, unlimited source of manufactured islets, and having enough for both the research community to test and develop these new strategies for protecting and delivering cells, but also potentially the opportunity to advance cells like this into clinical settings. All right, so next I wanted to highlight 2 new funding opportunities from our portfolio. Sort of what's coming up next and what's right at the next set of our pipeline. So the first is artificial intelligence powered approaches for T1D cell therapy. And artificial intelligence, machine learning has gathered a lot of attention recently in the biomedical research space as these techniques are sort of maturing and the applications of them are becoming further reaching. And so what we want to do is make sure that these advancements and these new technologies are being applied to the most important questions within type 1 diabetes, and in this case, specifically for cell therapy. And so as I mentioned, this is a technology-focused request for applications, meaning we're not focused on one particular scientific area, but rather focused on applying techniques like artificial intelligence and machine learning to either execute projects that couldn't be done without them, or to significantly accelerate the pace of research by using these tools. And so the 3 selected projects from this request for applications addresses multiple gaps within our strategy in this area, from better manufactured islet cell function and sort of benchmarking for comparing these cell sources, enabling survival at extrahepatic sites, so sites other than the liver, and enabling clinical translation for helping clinicians understand best decision-making practices when thinking about therapies like this with their patients. And then lastly, a funding initiative that is currently in progress, advancing technologies to enhance engraftment in cell therapy for type 1. And the goal here is really to keep pushing these technologies that can support the engraftment or survival of these insulin-producing cells at sites other than the liver. And so we know that proper engraftment and the survival of these cells is going to be a core component of any generation cell therapy, whether they're protected by immunosuppressants now or sort of our next generation strategies coming online. Focused on a number of research questions within this area. And I'm happy to say that with the projects that are currently under review, we had a lot of great reception for this. We have a high volume of applications covering diverse strategies towards enhancing engraftment. And we'll be prioritizing late preclinical studies that have the most promise of getting closer to the clinic. And so for the next section, I'm going to turn it over to Esther, who will cover the next portion of our call.
So thank you, Nick, for providing this exciting update on our portfolio. And I'm going to mention Project ACT, or Project Accelerate Cell Therapy. So as I mentioned, it's not reinventing anything on cell therapies, but it's an initiative to dramatically speed cell therapy products as T1D cures through coordinated efforts to simultaneously advance research, development, regulatory access, and adoption. So if you go back to the pipeline that I mentioned before, is actually accelerating the pipeline by all these teams, internal teams coming together, collaborating very closely with external teams and partners, and also the T1D fund. So why now? So I think I mentioned a little bit that it's it's why things are moving, our therapies are moving into clinical trials. And I think this is very exciting. For a long time, cell therapies has been relying on animal experiments and animal models, and sometimes not predictable. So we have the opportunity to learn a lot and advance pretty fast now moving forward. We have the first manufactured cells showing that it can cure people living with type 1. We have some preliminary data showing that gene editing as a strategy to protect the cells work. So we need to move to clinical trials. We need to support adoption of these therapies, as I mentioned. We need to make sure that that we are aligned with FDA, and we can work closely with regulatory, not only in the US, but abroad. And Breakthrough T1D is uniquely positioned to accelerate these therapies, and most importantly, immune suppression-free versions. So that's what we're trying to do, that even we're not going to move away from discovery. I think this is key to us to innovate and bring new therapies, but we still need to focus on accelerating first generation products, or testing these products in first in humans clinical trials. So I think I'm just gonna give it now to Mariana, my colleague. We worked very close for many years in the regulatory team. So we've been writing some of the guidelines together. So it's my pleasure to introduce now Mariana. Thank you.
Thanks, Esther. Hi, everybody. I'm Mariana Marinac. I'm with the Regulatory Affairs Group. So I wanted to bring this slide back that I know you saw just a little bit earlier, just to talk about the advocacy role as we work across this pipeline. Our goal here is to build pathways that ensure access to T1D cell therapies for everyone who is eligible and wants one. Breakthrough T1D is a unique organization in that we have staff in research, advocacy, and now medical affairs who works across this entire pipeline. This model was critical to the success of the Artificial Pancreas Project, which accelerated the development and availability of AP systems that many of us with T1D and caregivers are benefiting from today. And now with Project ACT, what we want to do, and we plan to leverage that same model and successes, working across this pipeline, advocating for more funding in terms for research and development, and policies that support that research and development. Accelerating regulatory pathways, which I'll talk a little bit more about in the next upcoming slides, and advocating for policies that ensure coverage and access, and educating our healthcare providers so that the same successes that we saw with the Artificial Pancreas Project can be seen for cell therapies as well. So let's talk a little bit more about accelerating regulatory pathways. The regulatory affairs team at Breakthrough T1D works with many stakeholders across the pipeline, including stakeholders like researchers in both academia and industry, policymakers like regulators, and also patients and caregivers to help identify barriers and gaps and eliminate them. Let me give some details on things that we're working on as part of Project ACT to help accelerate regulatory pathways. These efforts were built upon conversations that we had with stakeholders to help us identify gaps. And these are some of the things that we're working on to help eliminate those barriers. So one of the things that we're working on is a roadmap for the clinical trial design considerations for those next and future generations of cell replacement therapies in T1D. What you'll see in this roadmap that we're working on is our vision for those next generation of cell replacement therapies. And one of the key things that's important for us here is that we're expanding the patient populations with T1D who get studied in these clinical trials, looking beyond what we've been doing for the past 20 years and studying people with severe hypoglycemic events. We want other considerations to be brought in to help expand the patient populations that could be studied in future generations of cell replacement therapies. We have a manuscript that's been submitted, and we're doing some revisions right now. And we're looking forward to using this as a tool as we advocate to regulators and to researchers, both in academia and industry, so that they're thinking about representing and designing clinical trials that represent a broader T1D patient population. Similarly, there's two other really important aspects when a drug has to get reviewed and improved. And those are the animal studies that get done, as well as how you make your drug or your cell replacement therapy. And so the goal here is for us to provide a roadmap for non-clinical study design considerations and manufacturing considerations so that we can expedite and accelerate the development of cell replacement therapies in T1D. Non-clinical studies are really critical, a step in being able to conduct what's called a first-in-human clinical study. What these animal models can show us is establishing the safety of the cell replacement therapy and early proof that the therapy has the potential to work. We've spent a lot of time and resource funding research in this space, and so what we want to do is make sure that the community has a roadmap for what animal models can tell us about the cell source and the protection strategy and where we might be able to put those cells so that they can work best in people with T1D. Now, cells are also, they're living things. And so how you make them is super, super important. And we have this saying in the regulatory space, especially when it comes to cell replacement therapies, is that the product is the process. And so what that really tells us is how critically important manufacturing cell therapies are to make sure that the products are safe and that they can work. And so we're writing a roadmap. Again, what are the considerations as you develop a cell therapy from a manufacturing perspective that can guide our community? So we're in the process right now of writing these roadmaps, and we're looking to hopefully have those completed by the summer of 2025. And then we'll continue to work and use these as tools to advocate to the researchers, both in academia and industry, and the regulators and policymakers to implement these considerations as they're developing policies for cell replacement therapies. One of the key pillars for advancing and accelerating regulatory pathways is making sure that the regulators are up to date on the latest science. And you heard Esther and Nick talk about the beta cell replacement consortium and all the exciting and wonderful work that's going on in there. And so what we've done for probably over a decade now is we've hosted with the FDA a seminar series where we bring researchers from the beta cell replacement consortium to talk about their work specifically in type 1 diabetes. This is where we make sure that T1D stays front and center, and that the latest and greatest science and advancements are top of mind for the reviewers at the FDA who will eventually see some of these products come across their desk. This is where, this is the topics that we have coming up for our seminar series in FY25. And you'll see we cover a whole lot of aspects. We'll talk about patient perspectives on risk tolerance for cell therapies. And then some additional presentations from scientists that Breakthrough T1D funds about how to protect cells that could be transplanted. How do we create sites or an environment that can support cells that we transplant and that we can support transplanting. We also have what's called, and we're funding some work, what's called a patient preference study. And what this does, and really what we're trying to understand, is what's the patient perspective on risk tolerance for cell replacement therapies. There is a part of this study that I was also looking at disease-modifying therapies. So we're looking at early parts of the disease, as well as people who have had type 1 for quite some time. And really what we want to understand is bring some data and quantify what patients think are the benefits and risks for cell therapies. How we do these is we develop what's called a discrete choice experiment. And then you'll see this is an example, not from this study, but from one that we've done in the past, where you put these, you put together attributes, risks and benefits of a potential therapy. And then you ask people with type 1 and their caregivers as well, which one would they pick? Or which one did they think is the most beneficial for them? So we're just getting this study started. And we're hoping that by the middle of this year that we'll have more results to help us inform what are patients' perspectives and caregivers' perspectives. So we can use that information when we go talk to policymakers like regulators about what the unmet needs are for people with T1D and what kinds of risks and benefits they're looking for. And finally, one of the things that we heard as we talked to stakeholders in the cell replacement therapy space is that patient-reported outcome measures are really important to developing therapies. And what a patient-reported outcome is, it's something related to health or illness that's uniquely or best talked about or described by a person who has that condition. And really what these measurements do is they help us to understand how a person with a condition or using a treatment is feeling or functioning. They can talk about the symptoms that are related to the condition that they have and how they're able to function or their well-being in their day-to-day lives is being affected by the condition. Well, why this is important is that we don't really have good patient-reported outcome measures that can be used in developing cell replacement therapies. And that can support regulatory decision-making. There are benefits to potential therapies that might not be captured by traditional endpoints, like your hemoglobin A1C or how many hypoglycemic events you're having. And so what these instruments do, these patient-reported outcome measures, is they give us a look into what are patients' perspectives and what are the things that we might not be measuring from your blood or from a test that are important to people in terms of how they feel and they function, so that can be used as part of the decisions on whether or not to approve the therapy. So our impact in this field over the past 20 years has been tremendous. From isolating and transplanting islets from deceased donors, to differentiating cells into insulin-producing cells, to protection strategies like encapsulation, and even funding research into less toxic immunosuppression approaches. We've been steadily guiding this this field and this space. The launch of Project ACT is aimed to accelerate the development of cell therapies that don't require immunosuppressants. We're going to be working on accelerating those regulatory pathways by developing roadmaps, addressing the gaps that were identified by the stakeholders in clinical trial design, non-clinical studies, and manufacturing considerations. And then we want to also make sure through our efforts around patient preference studies and patient-reported outcome measures that the patient perspective on risks and benefits is part of the incorporated into the design of clinical trials, but also used by decision makers like regulators as they think about risks and benefits for the next generation of cell therapies. Thanks so much for listening today, and we're looking forward to the discussion.